Very good morning. Priti is there, Ante is there, and Richa is there, and gradually everyone is coming up. And today, the topic which we are going to discuss is molecular aspects of hormonal control of reproductive growth and the development. Okay, this is a bit kind of interesting, you can say, uh, because we have till now we have been talking almost all about the individual hormones and interactions and all, and from starting from the callus formations to the root initiation till the full ripening and the senescence. Today's topic is a little bit away from all those. You can say it's a clip, it's a basic point from among all those. That means mainly we will be focusing on the reproductive growth and the development, and how these hormones are really going to help during these reproductive, what are those constructive contributions of this hormone, especially on this reproductive growth and the development phase of the plant. Okay. Yeah, this is the slide that is says that the plant reproductive growth and the development. We all know that what is the reproduction phase. Reproduction phase, we say that if a plant should have a potential to give to something to new offspring. Okay, so these phases is very crucial, and in the last class also we had discussed that reproduction can be sexually and asexually and all. In the last class we discussed all about the this propagation method through asexually, and today mainly the reproductive, when we say that it should be through the fertilization. Okay, that is from the sexual reproduction. And the reproductive growth, it says that this is the phases which started from just from the initiation point of floral I mean the floral initiation till the uh, to the fruiting till the end of the fruit ripening. This whole stages made up the reproductive growth and development. We all know that growths are totally quantitative, and the development is qualitative. Okay, so we cannot measure developmental. So in developmental phases. So in the developmental growth, reproductive developmental phases, what we generally see is the process that whatever happening from the initiation of floral organ to the fruiting till the ripening. Okay, this whole progression, this is what the development which we are going to discuss today. Yeah, and it says that, as just now I said it, from the floral initiation. Again, the here under this also, under this reproductive stages, there are various minor stages are there, starting from the changes of this sporophyte to the gametophytic stage. Okay, from the gametophytic stage till the formations of all the individual organs, and again the individual gametes, the male gametes and the female gametes. Okay, that is what we mean to say the sporophytic stages changing to the gametophytic changes. Starting from these floral, that organ formations, then the every individual gamete formations, the male gamete and the female gamete. In this diagram, you might be seeing here, because once after these formations of the floral organs and the formations of the individual gametes, the next stage of reproductive comes is the pollination, which is again followed by the fertilization. Okay, I hope you know the difference between the pollinations and the fertilization. If not, then I can again I'll recall you this. Here, this is the floral organs. We know that one is the female part and the male part. This pollination, sorry, pollination, when the pollen grains from the anthers, it breaks it into the on the tip of this stigma, and there the starting from the pollen of the pollen to the stigma, starting from that till it reaches to the micropyle here, that pollen germinations, elongations of its tail, moving down through all these style and coming till this micropyle. This is what the pollination process is. And these two process, pollination and the fertilization. Fertilization is what? It is the fusion of that two gametes, one male gamete and the one female gametes. That is the fusion. That's why we say that when we talk about the plant reproductive and the developmental growth, we have to go basically to the sexual reproduction. Okay, so pollination part and the fertilization part. So this together, starting from the sporophytic stages till the fertilization. These are all the reproductive stages. Okay, and how in the developmental phases, I told you this is a progression covering all these stages from the initial till the ripening. It shows that here is the floral development. Then when the flower becomes matured, it will open, it will get pollinated. Then again fertilization. Once the fertilization is done, the fruit gets set, and after fruit setting, definitely cell enlargement expands until the maturity, and here at the ripening. This is whole process, the progressed of the total reproductive phases. Okay, so now what are the roles of phytohormones in these reproductive growth and the developmental phases? We have already discussed, and every time we keep on discussing, we keep on telling that these phytohormones has a basic roles, suppose in auxins for the apical dominance, for the GA, in the cell elongation, etc., etc., etc. Okay, but how basically it regulates this very crucial phase of plant whole life cycle? We all know that these phytohormones are the endogenous chemical substances which has a potential to transform almost all the plant specific responses. Okay, and that's why we say that when we say that have a potential to transform to a specific responses, and these responses may be anything. Okay, maybe the flowering, maybe the budding, maybe the fruiting, whatever, maybe the ripening, maybe the senescence, whatever. These responses which we could see it with our naked eye. Okay, this whole transformation is due to some sort of chemical signal which has been transferred from one cell to the another cell by the phytohormones. Okay, so we say that in a very low concentration, it could work out, it could carry out all these performances. So this is how the phytohormones has the potential to control all the aspects of the whole plant growth and the development, and especially the reproductive phases till the fruit maturity. So this signal, passing of this signal, we have said that yes, this phytohormones are chemical signaling molecules which has a potential to transfer signal from one cell to the another cell. And these transferring of one cell to the another cell, there should be one receptor, right? If signal is one, if I pass you something, then you should have the potential to receive that. Same thing in that way. Any signal which will be passed through by this phytohormones, there should be some receptors so that it can recept it. And almost all these receptors are the kinases, okay, are the kinase enzymes, histidine kinase. Basically, all these are present bound to the plasma membrane. Okay, so the membrane, that is the every organelle are bound with the and every cell has its a membrane. So in that wall, in that membrane, only all these receptors are present, so that from the the signal passed from one signal can be received by the other cell. Okay, so it says the reception, and once it get received by the receptor, it should have the potential again to transfer, to transduce. That is what a transduction is, and then finally the responses. Okay, so this is how the whole process like this: recepting, and then transducing, and then final responses. That may be the flowering, that may be the fruiting, etc., etc. But one very important point to remember in these is that these signaling one, okay, there is lots of variation, and this signaling of these phytohormones are totally dependent on the quality and the quantity of the phytohormones, okay, which also again varies from time to time. That's why, for example, we say that the auxin and the cytokinin, which really involve in the root initiation, is shoot initiation during the propagation time, plant propagation time. And again, we know that cytokinin in the cell divisions, auxin in the apical dominance, etc., and then GA in the cell elongations, internodal elongations and all. This same thing, how it is going to help in the this one flowering. This all says that yes, it is totally depend the signal receiving capacity of the signal, and also its responses is totally depend on the time to time, and also the phase of the plant growth and the development. It's very much dependent on these two, the time and the phase of the plant growth and the development. That's why during the initiation time and during the germination time, the hormone GA acts something else. It acts as a germination hormone. And when it comes to the flowering, it acts as a floral phytohormone. Like that, they change as per the plant phases changes and time to time. Okay. So next is the coming up to the individual phytohormones. The first one, auxin, I'm taking up here. How auxin is controlling, how auxin regulates this reproductive growth and the development of the plants. So we say that auxins, it has a varied role in whole aspects of plant growth and the development. We all know that. But subsequently, when we see particularly to the fruit, to the reproductive stages, it does has a, see, auxin specifies the site of floral initiation, and the second one is the organ development, organ growth. Okay, it regulates two. In the mind is okay, the auxin's role in the reproductive growth and development. One is the floral initiation, and the second one is the organ growth. So these two, how it is controlling, I'm coming up gradually. So to perform all these in a very better way, this auxin performance, to see this auxin performances, we came to know first from the isolations of the mutants from the Arabidopsis. Okay, and its action development have not been well defined until the recent identification of auxin biosynthesis mutants. Before that, we were just knowing it as auxins in other actions, but we are not exactly knowing that yes, this also plays an important role in the reproductive stages. So here with these, the auxin transport mutant, that is the pin1, this pin1, it get isolated and characterized, and it shows that this pin1, the full form of pin1 is also given here, is the peptidyl protyl cis trans isomerase NEMA interacting 1. Okay, so when we say that when there is a, whenever this mutant is there, I mean, it can be the it has been experimented that auxins, the, if there is an auxin, if it's a normal plant, then there is a flowering. But in the mutant plant, where there is a presence of this pin1, it shows that, I'm show here, yeah, here it is. Okay, it shows that there is no flowering in this. There are also other mutant, that is the pinoid and the MP genes, where these are also present. This also shows the same phenotypic responses as that of mutant plant with the pin 1. Okay, that means no flowering. And this one, it is the wild type, where there is no mutant genes are present in these, the flowering is normally coming up. Okay, so it says that because of this mutant, flower flower induction in the plant is been reduced or is been blocked. Okay, so it says that the pin1, why we have given the term pin here, pin1 mutant displays pleiotropic defects throughout almost all stages of development. But the most striking phenotype is that the inflorescence of pin1 often does not have any flowers, suggesting that auxin plays an essential role in initiating the floral primordia. That means floral initiation. Okay, so why this pin type has come up, as we know that when there is no flower, almost all the plants, they just show a naked state without any flowers, and it just like an arrangement of a pin in any of the this one medium. So that's why from that the term has been drawn, the pin. Okay, the pin one. So it says that the mutant plants, the inflorescence produce are pin like. Because of that only we say that it is a pin one gene which muted the plants and not letting the plants to get flower. One more thing is that this mutant pin one gene, which encodes the PIN1 protein, function in polar cell to cell transport. We know that auxin has a polar transport, yes, polar movement, precipitally from the apex to the basal part. And here it says that by regulating coordinated influx and the efflux of auxins into and outside of the endoplasmic reticulum. Okay, so formations of flowers is triggered by regularly spaced local auxin accumulations at the surface of SAM. That means auxin's presence only in these shoot apical meristem. Okay, then only the flower will be revived. And this protein, which helps in this polar movement of the auxin, is being encoded by the mutant gene PIN1, and because of that the flower is reduced and there is no flower at all in the flowers. And this all proves that yes, auxin is also involved in the floral development and also in the organ growth. Now, floral initiation one we had discussed it. What about the organ growth? Okay, here it says that auxin is required for fruit growth and development and delays fruit senescence. We know that where there is cytokinin and auxin, fruit senescence get delays. Okay, by acting antagonistically with that of the ABA and the insulin, which enhance the fruit senescence. Okay, so and these are the examples that they just see it by removing this, because seeds are very seeds contain a higher concentration of auxins. So they have experimented in the strawberry by taking out the seed from the strawberry, then the growth of the strawberry stopped there. Okay, and again we apply it auxin exogenously, then the fruit size increases. It says that yes, auxin is also involved in the fruit growth. How molecularly it is? Here two things are involved. Okay, you can see this. This is the transcription factor TIR1. This is, you know, the transcription factor which enhance in these transcriptions. Okay, and one is the ARF. ARF is the auxin response factor. In any of the plants where these auxin response factors are present, these fruit growth will be increasing. Okay, but what happened to you just see in these pictures now? One is minus auxin, that is absence of auxin, and the presence of auxin. In the absence of auxin, what it does is that this transcriptional factor is let as free, and this is the repressure one. Okay, this auxin oblique IAAS, this is the repressure of the auxin response factor. It represses these factors, so there is no auxin response. That means no fruit growth, no flower initiation. Okay, and in case of auxin, where there is auxin is there in the plants, then this transcription factor TIR1, it binds to these repressor molecules. Okay, it binds to these repressor molecules and not letting to bind with the ARF. That means in the presence of auxin, the auxin transcription factor bind with the repressor, and the repressor could not again could not anymore hold this response factor. So that as the auxin response factors are free, there are changes in the transcription, and so the floral initiation and the fruit growth are maximized in this. Okay, and this is how the auxins involved in floral initiation and the fruit growth. That means the whole plant reproductive growth and the development. Okay. Now, GA, the role of GA again. We know that GA is one of the hormone. You might have, if you remember the photoperiodism, GA really plays an important role in that. Okay, here GA induced the flowering in the long day plants in the short daytime. Okay, so during this short day time, it induced the flowering in the long day. Here it says that GA enhance long day flowering in short day conditions. Due to how it has been controlled, due to the regulation of, note this, okay, for the GA, regulation of GA20 oxidase gene expression. There are so many GA20 oxidase, GA30 oxidase, like that. So here the GA basically among all the GA20 oxidase gene expressions plays an important role in inducing of these flower in the long day during these short day conditions. Okay, and these signal always comes from the leaves to the SAM, where the floral initiation will be taking place. So now in the mutant plants where GA13 is present, like same in that of auxent, okay, where the pint 1 is present, pinoid is present, MP is present, there is no flowering. Like that, the same thing here in the mutant plant where this is the mutant gene, GA13, this is present, then there is no flower initiation in the long day plants, because it check the GA biosynthesis there. But suppose if there is no biosynthesis, if there is not at all GA, then there is one more interesting uh, thing is coming up here there which can substitute GA. Okay, that is the constant gene. In short, we write as a CO. Okay, this constant gene has the potential to substitute this GA1, and even even though in absence of GA1, if constant gene is present, then it can promote flowering to the plants. Okay, so now it says that the GA promote flowering through activations of gene encoding the floral integrators like SOC1. The full form is suppressor of overexpression of constans 1, leafy LFY, a floral meristem identity gene, flowering locus T, that is FT, in the inflorescence and the floral meristem and in the leaves respectively. They all enhance this, okay, so that the flower may come up in the plants. So what did genes generally encode? Proteins that activate the floral meristem identity, that is the FMI, genes like the R petala 1, in short we write AP1, R petala 2, AP2, fruitful, FUL, and in cauliflower they have isolated these and characterized, so it's given the cauliflower and CAL, and LFY, that leafy, okay, which convert the vegetative meristem to floral fig. That means because of by enhancing all those genes will only how by enhancing only doesn't how it promotes the flowering. That means by letting these vegetative meristem to convert it, to change it to the floral meristem, so that the flower initiation can generally come up. But one more interesting thing again here, what in absence of ZA, and what if there is no constant gene present in the plant, whether that plant will be flowering or not. Okay, here this is controlled by one of the protein, that is the DELA protein reset. Okay, in absence of GA, DELA protein, what this is, come to this one, this picture. In this diagram, what we see is this is the chain where the transcription factors will be coming up and bind to the promoter and will enhance this gibberellin responsive gene, and that's how the gibberellin action will come up for the flowering and the fruit and for the fruit growth. So here what they do is that when there is DELA Okay, in absence of GA, the transcription factor, see here, the transcription factor comes and bind to the dela. Okay, in absence of GA. In absence of GA, this transcription factors, what is the function of this transcription factor? This transcription factor should come and bind to this promoter so that it can initiate the chain. Okay, but as there is no GA present in these transcription factors come and bind to the dela, and dela is not letting these transcription factors to go and bind with the promoter to initiate the chain and enhance the gibberellin responses. Okay, but in presence of GA, what we do is what it generally happen is that the transcription factors remain free. Okay, in the presence of GA, transcription factors remain free and it's not binding with the dela. Instead, it goes to the promoter side. Then what happened to the dela now? Dela is already present there. So what happened to the dela is that GA along with this GID1, okay, it binds with this and then it degrade this dela. Okay, in the presence of GA along with this GID1 bind with the dela and the dela degradations uh, took place at that time, and so the transcription factor is free, and as the transcription factor is free, it can easily come and move and bind to the promoter sites, and that's how the gibberellin responsive genes are induced and finally to the floral initiation. Okay, so gibberellin regulations of fruit set and growth in the tomato. Pollination mediates fruit set by activating GA biosynthesis mainly through upregulations of GA20 oxidase again. If you remember, I don't know how many of you were there in the 508 in the last semester, and those who were there, they might be remembering it in the fruit developmental, fruit set and the fruit developmental uh, processes, GA's role in that. During the pollination, that's why I say that for any reproductive, these pollination and the fertilization have a very crucial role in the whole reproductive growth and the development, because at the moment of the pollination the GA action raise up in this GA action raise up and it started enhancing the seeds the whole seed development. Okay, so that is why it says that here gibberellin plays an important role in fruits, and not only in the tomato. I have given the examples in tomato. Here in other fruits also it is enhancing the process is same, and the main regulated gene is that GA20 oxidase. Okay, now coming up to the cytokinin. Cytokinin, as we know that, yeah, coming here. For the cytokinin, as we know that the a cytokinin has an important role in the reproductive growth and development again, because how we know that cytokinin is the one which help in the cell division and also in the cell enlargement along with the other hormones like GA and the auxins. Okay, so it says that cytokinin regulates the activity of reproductive meristems, floral organ size. That means almost everything same as that of the auxin and the gibberellin did. Okay, means it has the same actions. It also promotes the floral initiation and also the fruit growth and the development. Okay, so cytokinin has a central function in regulating the activity of the same SAM, the shoot apical meristematic, that zone. It really control the SAM, where the floral initiation will be started. Okay, so it says that in the reduction of the cytokinin status, which has been achieved either by lowering, means we just now I have said that the cytokinin promotes the SAM region, and that's how the fruit formation and that's how the floral initiation is there. But to experiment this whether exactly the cytokinin does involve or not, there are two things that we reduce the cytokinin action. Okay, in two ways. First, either by lowering the cytokinin content or by reducing its signaling. That means to pass, okay, to pass a signal or by lowering its content. When we do this, it suddenly the SAM region get concise and there is no floral initiation in that. And that's how it proves that yes, cytokinin is a positive regulator of SAM activity. Okay, so this SAM, that means is totally dependent on the cytokinin. If there is cytokinin action, floral initiation. If there is no cytokinin, no uh, uh, this one, no activity in the SAM and no floral initiation. So this is again the cytokinin contents. It's again regulated by the enzyme cytokinin oxidases and the dehydrogenases. In short, we write the CKX. S means so many. Okay, so it can be CKX1, CKX2, CKX3, 4, 5, 6, so many are there. Among all these enzymes, the cytokinin dehydrogenases 3, that is CKX3 and CKX5, really involved in the reducing the floral initiation and at the SAM region by degrading the cytokinin concentration. Okay, so this is how we say that SAM is totally dependent on the cytokinin concentration present at the SM region. There are few more genes like GN1A, locus of rice. This is an example in the rice which make a major contribution to the regulation of grain yield, carries a mutation in the promoter region of the OSCKX2 gene that reduce expression of CKXY. That means if this gene is present, okay, if this gene like constants in case of the GA, like that if this gene is present, then it reduce the expression of these dehydrogenase enzymes which degrade the cytokinin, and that's how they protect the concentration of the cytokinin and promotes the SAM reason for the floral initiation. Okay, now roles and the regulation of cytokinin in tomato fruit development. Besides auxin and the GA, cytokinin is also known to induce fruit set in several fruit crops. The endogenous level of cytokinin is directly correlated with the fruit growth, especially in stimulations of cell division. Okay. A very interesting thing here again, there are so many genes involved when we talk about the cytokinin and its involvement in the fruit development, in the fruit growth. Okay, and these also in two ways it acts and involving different regulatory genes. And the genes, these are the genes which directly enhance and induce the cytokinin biosynthesis. So when these genes are there in the plants, the cytokinin biosynthesis is there and the ovary growth is there. Okay, me here in the two ways I said the one way is the cytokinin works in the unpollinated ovary and another one in the pollinated ovary. Okay, so when the first one, if we consider that is the unpollinated ovary, what it does is that when these genes are present, it enhances the cytokinin action and promotes the ovary growth there. So that's how definitely the fruit growth, it has been controlled by the cytokinin directly with the involvement of these genes. But what about if the pollination takes place? Okay, so in the pollination takes place, then once the pollination happen, then these genes comes out and these gene replace the cytokinin action. Okay, one gene get induced at the time of pollination, the second one Under the unpollinated one, directly controlled by these genes, is biosynthesis of cytokinins and help in the ovary growth. But when these flower get pollinated, then instead of these genes, they induce this gene, that is the SICKX17, and these gene repress the biosynthesis process of cytokinins and not letting the fruit grow. Then what about then how the fruit is going to grow if there is a pollination? The answer of that is that again, once the pollination, it goes to the three ways. One, induction of other genes rather than this one, that is the 4 and the OG6 and the 8. Besides it here, these genes will get induced and these will be acting on the transgeatin, which further led to the pedicel growth. You know what is pedicel? That the flower and the fruit stalk. Okay. And this one which finally lead to the fruit growth here. And one more is that once the pollination is there, we know that GA action is increasing. Yes. And here the cytokinin from the transgeatin, this is still unknown, this pathway, but probably it has been believed that it has a control on this oxygen action, which further help in the cell division, cell expansion, and later on the fruit development. You understood now? Two things here. One is the when unpollinated ovary, and another one is in the pollinated ovary. In the unpollinated ovary, induction of the genes direct fruit growth. And when the pollination is there, three ways. One from the transgene and one from the GA, and the third one, the transient control over the oxygen, which further led to the cell division and expansion and then to the fruit development. This is how again the cytokinin is involved in floral initiation and fruit development. Okay. Now the next hormone, that is the ethylene. This is the fourth hormone we are discussing now. How we know that ethylene and the ABA, they are the retardants. Yes. So they are the growth retardants. Definitely, as it says, they are going to act as an antagonist to that of the GA, to that of the oxygen, and to that of the cytokinin. Here again, the ethylene, what it generally does is that it regulates the GA biosynthetic pathway by regulating the DELA protein. You remember the DELA protein which just discussed in the previous slide, how the DELA protein is there, bind with the transcription factor, not letting the transcription factor to go and bind with the promoter, and that further, which will further enhance the gene expression, that GA responsive genes. Okay, so here how the ethylene does is that it regulates the GA biosynthesis by manipulating the DELA protein. Okay, so it says that ethylene enhances DELA accumulations, in turn delays flowering, that is through repressing floral meristem identified genes, that is SOC1 or LFY. Okay, that FMI and all the whatever which that inducing genes in the presence of GA that enhance the flowering, all these get repressed uh, by giving by enhancing more accumulations of DELA in the SAM regions. Okay, so in absence of ethylene, what we see is that ETR1, it has activate another CTR1, a serine kinase that negatively regulate ethylene signaling, and GAGID1 works on DELA. That means we see the experimenting whether it is really DELA is been manipulated by the ethylene and all. So the ethylene get, in the absence of ethylene, there are few genes which get activated, this ETR1, and that further activates the CTR1. And these, sorry, these act, negatively to that of the ethylene biosynthesis process and enhance the GA activities, that the binding activities with that of the DELA, and making the DELA protein degraded. Okay, so it says that downstream means reducing of the CTRI regulates expression of ethylene response with transcription factors EIN2 and EIN3. That means this ETR1, which promotes the CTR1, then the ethylene biosynthesis is down, thus enhancing the GA biosynthesis process and also its activities. But when this CTR1 is so much reduced, so much down regulated, then what happens? So many other transcription factors are coming up, that is the EIN2, EIN3, etc. These are the transcription factors which promote the ethylene biosynthesis and thus enhance the ethylene responses by not letting the GA to come over and take for the flower and the fruit initiation. Okay, so it says that the stability of EIN3 deactivated ethylene actions. We have to manipulate in these transcription factors which really reduce the ethylene actions. So another example is there that in the malons, in this cucurbitaceae family, the CMAC-S7 and the CSACS2 of malon and the cucumber respectively regulate the arrest of stamen development. Mind this. It manipulates the stamen development in the female flowers of monoecious cultivars, and their loss of function mutations lead to the conversion of female into bisexual flowers and therefore the transformation of monoecious into andromonoecious cultivars. Monoecious to andromonoecious. That in the monoecious there is one female and the one male flower will be there in one plant, right? So here all these female plant will get female flower will get manipulated, and what happened? These monoecious plants turn out to be andromonoecious. Andromonoecious means one female flower plus the hydro that and the bisexual flowers, hermaphrodite. Okay, so two sex in one flowers and the one male. That's why we said that from the monoecious it get transformed to the andromonoecious plants. So ethylene genes are highly expressed in ovary of tomato flowers and anthysis, especially in parthenocarpic genotypes, but they are downregulated after fruit set, which suggests that ethylene act as an antagonist to auxin in fruit set, preventing carpel development into fruit before pollination and fertilization. That means from the onmark, from the before the onset of all this pollination and fertilization, this ethylene action already checked the formation of these already checked. That's why there is reduced and there is no formation of fruit and no fruit set in the plants.